Hi, I'm James Graham, CEO of ASX-listed Recce Pharmaceuticals Limited. If we jump through our slides, we have the usual disclaimer, and here we have our team. Our chairman is Dr. John Prendergast. Dr. Prendergast is chairman of New York Stock Exchange biotech company Paladin Technologies and lead director of NASDAQ biotech company Heat Biologics. His doctorate is actually in the study of bacteria and blood diseases, and as you'll hear, as an infectious disease company, that's very important to us. I was previously, for some 5 years, the executive director as a member of the board, which I've recently transitioned into the position of managing director and CEO. By way of background, I actually started as the founding investor of this company. I find business interesting. If you look up my name, I've invested in all sorts of stuff on the side, but really my investment was fundamentally supporting capable people. And those two particularly is Michelle Delizia, who's our chief scientific officer and co-inventor of Recce anti-infectives, and Dr. Graham Melrose, who was former head of research of Johnson & Johnson Australasia and executive director of their board for about 10 years. When we look to where that technology is transitioned to, we really consider that from a position of an umbrella as an infectious disease company with two pillars, antibiotics and antivirals. The commonality between the two is because, of course, you shouldn't ever use antibiotics on viruses, is the binding and interaction with the proteins of the cells. So the outer cell of bacteria or the envelope of the virus itself. That's what we believe we bind to and interact with. We currently have two clinical programs in the background. Our first program is a burn wound infection program. So it's really utilising our technology as a spray-on antibiotic on the vulnerable burn wound itself. It's a phase 1, phase 2 study out of Western Australia, led by former Australian of the Year and a global burns expert, Professor Fiona Wood. We would expect interim human data to be available in the very near term, and we certainly look forward to communicating that. In parallel to that is our sepsis program, so utilising that same solution but as an intravascular solution, tackling the bacteria that exist in blood. Basically, sepsis is blood poisoning as a result of bacteria entering, and as we see further in, it's the #1 most expensive condition treated in health. Outside of that, we have the traditional preclinical programs, moving them through the usual safety efficacy type studies as IND enabling pathway for the purposes of or intention to bring them on as other clinical candidates. This is it as a traditional snapshot, and there, of course, we see near-term interim data in the top two regards there. So sepsis, the #1 most expensive condition treated in health, double the second most. It's a major unmet medical need. There we see around 50 million persons each year contract sepsis, and 11 million of those 50 in fact die, or in hospital, a 1 in 3 death rate. And the reason is there's currently no antibiotics specifically for the treatment of sepsis. So why is that, or what's the clinical challenge? The challenge is the treatment paradigm. When a patient presents at the hospital itself showing signs of sepsis, the clinician quickly draws blood, rushes it off to the lab where they cultivate that blood out to try to determine what type of bacteria is in that patient's blood and what type of antibiotic is that bacteria susceptible to. That takes time, and for every hour left untreated, the likelihood of survival decreases by 8% each hour. Now, our antibiotic is designed with the end in mind, to avoid that time-consuming guesswork, which currently they'd administer a cocktail of antibiotics in pure hope that the bacteria responds. So the faster you get into the blood system, where in our case, we're attracted to the proteins of bacterial cells where we bind and interact with, the likely better the patient outcome, as we've seen in all of our studies to date, and we encourage you to look through our announcements accordingly. We believe that would eliminate that guesswork, and of course, getting on top of patient infections leads to better patient outcomes. All existing antibiotics is naturally derived. So you're taking bacterial fungi, cultivating them out, and you're putting them against, essentially as an antibiotic against the offending bacterial fungi. But you're only as good as what's found in nature. And bacteria will continue to mutate. It's a survival mechanism, and that traditional mechanism of action no longer works. Well, beginning with the end in mind, we've designed ours to have multiple mechanisms of action, and particularly to be attracted to gram-positive, gram-negative bacteria and not care or not discern about their mutation process, as we see in our mechanism of action studies. This is actually talking a little about that. So we basically have an initial attraction and binding to the bacteria itself, whether a gram-positive or a gram-negative, in this case expressing E. coli. We enter or permeabilize the bacterial cell wall, where when in the cell itself, we interact with the ATP, and the ATP production is effectively the beating heart or the lifeblood of the bacteria itself. We shut down that beating heart or that life that the bacteria itself has, and we shut it down irreversibly. That's the first mechanism of action. The second mechanism is we actually stop the cell from dividing or the infection from spreading. There's another mechanism. And as we actually see jumping across to stage 4 there, we know when in higher concentrations, we take advantage of the metabolic processes which create intercellular pressure to cause bacteria to burst. No other cells in the body have that high internal pressure, and that gives us a high degree of selectivity amongst the other activities happening here, by example, to target and interact with the bacteria itself, which of course is intended to lead to better patient outcomes. This is really a detailed explanation of it, and I think we really did it well in a recent announcement and an abstract that was released in a recent conference. This is looking at a molecular level, of course, the buoyant balloon-like bacteria bursting, and of course, no more bacteria, no more problem. ESKAPE pathogens is an acronym that represents essentially the the bacteria that are responsible for 45% of all sepsis or bloodborne infections. And what we're really looking at here is how well do we work or discern between a natural form or a superbug form. When we think a superbug, we think a super strong. You'll need a lot more concentration. You'll need a lot more exposure and all the rest of it. Well, actually, due to our mechanisms of action, when you look across these different kills of the bacteria, in many instances, we actually sometimes work better against the hypermutated superbug form. So briefly, we see the first one there. The blue is the standard, everyday form. The red is the vancomycin or drug-resistant superbug. Well, we actually worked better against the superbug than the standard form. That's nice to see. And then, of course, when we look across as a general snapshot, sometimes better against the standard, sometimes better against the superbug. But ultimately, we don't care whether it's a standard form, a superbug form, a gram-positive, a gram-negative. It's just a bacteria to us, and it's that broad spectrum capability that I believe represents a lot of value above and beyond existing antibiotics to date. Does it work quickly? Well, yeah, there we see a significant bacterial reduction in all of the represented bacterial types there. It's actually what's called a BLOQ, or below limit of quantification. Does it work and keep on working with repeated use? A fundamental challenge of all existing antibiotics to date? Absolutely. And here the commercial antibiotic, being amoxicillin, stops working after only a number of repeats. This is our first human clinical trial in the background, which is focused on sepsis. It's a safety study, meaning the people are healthy, and it's basically a dose escalation study of how much of the compound can a person reasonably take in the blood. We've obviously done that many, many times in animals, and we seek to dose escalate to effectively find the ceiling with the intention of the therapeutic window between where it works against the bacteria and where there's too much compound administered into the blood. That's pharmaceutical development, and we very much look forward to the various cohorts mentioned there. The very near-term clinical data result will come from this burn wound study. As mentioned, led by Professor Fiona Wood and fellow colleagues. It's sponsored by the West Australian Health Department, so it doesn't cost us a penny, and that's caveat-free to us in regards to the clinical data supporting our overall development. In the background, utilising this new class of anti-infectives, in this instance is an oral solution, so targeting Helicobacter pylori, which exists in the upper duodenum of the gastrointestinal tract. People often go, what about the good bacteria? Well, they exist in the lower gut or the lower intestine. And as our data to date supports or indicates, I should say, we either through our 100% water solubility across all pHs break down as it interacts with the bad bacteria in the upper duodenum, or breaks down due to that solubility. So when we get down to that good bacteria, we don't appear to have any existing antibiotic properties, which is a unique advantage in our case. So we're in the middle of our preclinical studies there, but we intend, with success, or assuming success, to bring on a clinical program thereafter, which it would be potentially at some time early next year. This is looking at a compound from an antiviral side. The Australian government selected us as a priority 1 test candidate in their COVID program. We're actually in stage 2 of 3 stages in that program. As far as I'm aware, we're the most advanced in that program. And this is just a snapshot of some of the data. There in the top side, we see a 99.9% efficacy. But looking at did we harm the healthy cells? Well, actually, no, the healthy cells remained healthy and viable or intact, indicating a specific interaction or a specific capability, targeted ability against the viral cells itself, which is, of course, a very good thing. Our intellectual property estate, well, it's really been a land grab opportunity when we look to synthetic antibiotics. Our family 1 is composition of matter, so manufacturing and curative claims. Family 2 is preventative, so preventing infections taking place and expanding upon our methods of administration. So intravascular, topical, oral, nasal, inhaler, so on and so forth. And we see obviously those granted all around the world. Family 3 is our antiviral candidate. So that's with granted claims for coronavirus or viruses, HIV, influenza, Ross River viruses and similar. Extending many, many years out from where we are today. And by example, of course, with our QIDP designation in the United States, we have government-backed market exclusivity, which comes above and beyond our patent position, which would start from time of approval. So patents can fall over. We're not at all concerned about anything there. But if they did, we have a government-backed market exclusivity unique to that sepsis program, starting only from time of approval. And we're thankful for that. We manufacture our compound under GMP specification. We've obviously provided our clinical samples to the various facilities around, and we do that as a method of synthesis, which is extremely inexpensive and more effective than a traditional method of cultivation, which is all existing antibiotics to date. We have a lot of cash, around 20 million cash. By example, our burns program, the phase 1, phase 2 doesn't cost us. It's WA government sponsored. Our intravascular program, well, it's about $2.5 million, doesn't cost too much. Our fixed monthly burn rate is about 150 to 200,000, oh, really, I say $200,000 a month. So really we have horsepower there. We have cash for the purposes of deployment, for the purposes of these preclinical assets moving into that clinical pipeline to deploy that capital for the purposes of advancing through the typical drug development pathway. There's obviously significant founder ownership. Behind a number of the nominee accounts are often one holder in our case, a large institution or institutions. Certainly there's a number of those within the top 20. So really as a summary, look, we're an infectious disease company in the midst of a global infectious disease crisis. We have the first new class of antibiotic in over 30 years. And this antibiotic having the near-term catalyst of indications to assess efficacy in a vulnerable and major patient population of burns, with, of course, the traditional sepsis safety intravascular study at clearly 2 leading Australian institutes. We're well funded. This is a global perspective of major patient populations, and we very much look forward to communicating news flow and opportunity as presents in the company along the way. Thank you for listening.
Thank you, James. Time for questions. We've got a fair few here, mate, so I'll rip into them straight away. Robert King, have you had permission for emergency use, last resort, of the drug against sepsis in Australia for a while now? Can you divulge any results?
Yeah, hi, Peter. Good to be with you. Look, under the Special Access Scheme managed by the TGA, there's Special Access Scheme Category A, I recall it to be, and I'll just have to double check that. But basically, if a clinician determines that a patient is unlikely to survive with existing antibiotics or existing drug therapies, and they really have to go to an experimental medicine in the best interests of that patient, that experimental medicine can be deployed. We actually had an instance in our case, as ours isn't particular to just sepsis. It's a condition that we obviously have a key interest in, but is certainly not the only area of unmet medical need, where we talked about a antibiotic-resistant a pathogen in a sinusitis case of a middle-aged male. That patient actually responded very, very positively. It's only a case of one in this instance, but that Pseudomonas infection, very drug resistant, in fact, multi-drug resistant, was an excellent outcome for the patient and the company as a whole.
Okay, question from Rob Beattie. So is this a broad spectrum antibiotic?
Most certainly, yeah. So bacteria are defined by gram-positive or gram-negative. When you work against both, you've got yourself a broad spectrum. And obviously, in our instance, we keep on working with repeated use. So broad spectrum is an indication of capability across multiple pathogens.
Okay, Robert Spitz. Not sure if I can ask this, he says, but I can see revenue is increasing, which is great. However, 2020 and 21 shows a large increase in expenses. 2019, 2.95 mil, I guess, 4.4 in 20, and 14.27 in 2021. Now, you're running this show now, James. Are you having a lot of parties there or something?
That's right. That's the beer and chips budget. We ramp it up year on year, but no, it's look, we're obviously a drug development business, and until products are approved for sale, there is no revenue, frankly. We have 43.5% R&D rebates that we receive on our programs in Australia. We quite uniquely to us have that extended to any R&D activity undertaken anywhere in the world. And really, as we've stepped into these key clinical studies, as noted in the background, we've had an increase in our expenditure. I noted we see money as a tool to be invested and utilised, and I see our near-term news flow events will be consistent with the activities that money has provided us.
Okay, Steve Bible, what is your expected time before it comes to market and is approved?
Depending on the program, look, I think the lowest hanging fruit or the easiest pathway to market would likely be the topical antibiotic solution, which is currently being utilised on burns patients in Fiona Stanley Hospital of Western Australia. I would see that probably still a couple of years from where we are today. It's a collapsed phase 1, phase 2 study, which suggests assessing indications of safety, whilst of course the upside speculative event of efficacy, multi-drug resistant pathogens and similar. But each step of the way, you obviously have a news flow event and a step towards that potential market approval. And if successful, then it'll be pleased, of course.
Okay, question from Judy McLaren. What is your key differential of existing antibiotics and antivirals?
Well, we work and we keep on working. I mean, from the perspective of antibiotics, I remember the day if you'd go to the doctor, they'd give you one prescription, say, take that and you'll get better. Now you go there and they say, take that and these follow-on repeats just to overcome the same infection. And the reason for that is the drugs don't work. Bacteria mutating, they always have, they always will, and with no new class in over 30 years to keep up with that, it really is, the perfect storm. And we've addressed a compound from first principles to overcome the challenges of nature and hypercellular mutation. And I think it is just that, by beginning with first principles in mind, that will deliver us, I believe, the result.
Okay, here's a question from Garonwe Price. I think I read somewhere you have already dosed a patient or patients in a critical condition with the antibiotic. Is this correct? And what was the result? Are similar likely?
We did announce in our burn study instance that multiple patients had been dosed. We'd announced in that instance that there was no negative side effects at that time. I have not received any additional data, but that was a couple of months ago or something similar. And we are all patiently waiting for the near-term clinical results to expand upon that previous position.
Okay. Well, mate, we are out of time. Thanks for joining us, and good luck with it. I think most of us would like to think that something like this would work. It sounds like it's like the Dalek approach to medicine: exterminate, exterminate.
That's it. Great to be with you, Peter. Many thanks.
Thanks, Matt. Thanks for joining us.
Cheers.